Hey everyone, today I'm going to be talking about enzymes and how they are kind of working in our body and make everything kind of happen. So let's start here with this awesome graph. Okay, so this is pretty simple graph, but very important. You'll see this a couple times kind of throughout the lesson here today. Um, Reactions occur, right? Every process in our bodies requires some type of chemical reaction to make it happen. Okay, enzymes we'll learn about here in a second. I talked about it with our macromolecules. Enzymes are a protein that kind of help these processes along. Okay, but in order for a chemical reaction to happen, you have to have what we call activation energy. Okay, so there's always some input of energy to make a reaction occur. It could be heat, agitation, another chemical, something like that that makes processes occur, chemical reaction to start. So you can kind of see in this graph, right? We have energy level before we input some energy there, and then at the end we have lower energy because the chemical reaction has happened. But that input of energy you can see is then what we are looking at here as activation energy. Okay. Two types of reactions I want to talk about. The first one is anabolic reactions. Anabolic reactions create larger products. Okay, so at the beginning, right, you have low energy, energy is input, and at the end you have more energy than what you started with. Okay, so here we are making larger molecules. Okay, we're taking small molecules and we're making larger ones. Think about adding two monomers together to make a larger molecule, a polymer. Okay, in this case, energy is required. Right, you can kind of see here, energy is required. We need that. Some heat or whatever is input. So again, looking at the graph, we see there's a ton of energy input. Um, at the end, there's still energy because that energy doesn't go away. It is now in the product. Okay. These are what we call endothermic or endergonic reactions, which means energy is taken in. They might feel cold, right? Because energy from the outside is taken in. Okay. So endothermic means the energy comes in or the heat comes in. Okay, so anabolic reactions are really important. We're taking two molecules and then creating larger ones. The opposite of that are catabolic reactions. Catabolic reactions are breaking down molecules. Okay, so you can see here energy at the beginning in our graph is high. We input that activation energy, and at the end we have a lot lower amount of energy. That energy is released into the environment. Think about breaking down food, right? So you eat something, you break it down. Now the energy is released. We get to use that energy for our bodies. Okay. Sometimes this might be like a gas in a car is a catabolic reaction. You're breaking down the gas, heat's released. You get to use that energy and that heat to drive your car. Okay, so they're also called exothermic or exergonic reactions as well. So here's a couple graphs, right? Number one, right? What is that type of reaction, right? So we see, it's kind of hard to see, but we got our molecules there, right? Energy is input, and at the end, two molecules are separated, right? So that would be a catabolic reaction. Oops, sorry. It'd be a catabolic reaction, right? Because we are releasing energy, right? Bonds are broken, two new molecules broken down, right? Over here on the right, you can see the opposite. We have two molecules separate from each other, right? Then at the end, two molecules are put together, so molecules coming together, energy is in there. That would be an anabolic reaction, okay? Let's talk about a couple other things now. So chemical reactions occur in our bodies naturally, right? A catalyst is anything that can help change the process, right? It will speed up the rate at which that chemical reaction occurs. Chemical reactions will just naturally occur in our body, right? Which is kind of cool. But we use catalysts, right? We want to use these catalysts. We want chemical reactions to use catalysts as well, because if we don't, what ends up happening is they'll take forever, okay? So what we have in our bodies are what we call enzymes. Enzymes are biological catalysts that help speed up and break down things in our body. Okay, so they help speed up these chemical reactions, either building or breaking down molecules. Okay, so for example, sucrose, if we want to break that down into glucose and fructose, right, we have this enzyme called sucrase that helps that process. Sucrose will just naturally break down, but the cool thing is we can add an enzyme to it and it will help speed up that process. Okay, and so two things enzymes will do: they can either speed up the reaction, or they can help lower the activation energy. Okay. So enzymes are catalysts that we use in our bodies. Most are proteins. We talk about that with our macromolecules, right? Proteins, enzymes, right? So these enzymes speed up reactions or lower activation energy. Okay, very common test question, by the way. Generally speaking, they are not changed by the reaction. You can use them over and over again. Okay, and kind of fun. They're usually named for what they do. Lactase breaks down lactose. Amylase breaks down amylose, which is starch, and so on and so forth. And usually they end in ASE. So if you see ASE, you know it's an enzyme. Here's what enzymes do, right? So again, here's reaction before the enzyme on the left. You can see it takes a lot of activation energy. The process is happening. Enzyme added on the right. You can see now there's lower activation energy. So very cool. Here are some that sped up the process, right? So lowered activation energy and sped up the process, and kind of both of those examples. So enzymes, right? Let's talk about how they work here. Okay, you have a substrate. Substrate is the thing you want to either break down or build up. Okay, it's the thing that will undergo the chemical reaction. It is going to bind to what we call the active site. The active site exists on the enzyme. See this picture, right? The active site is on the enzyme, right? The substrate binds to that active site, and then it will, hold on, and then it will, the chemical reaction will then occur. Okay, so the enzyme binds with the reaction, or with the substrate, kind of like a ball in a catcher's mitt, like it goes to it, and the catcher's mitt will like squeeze around the ball as you catch it. Same thing here with the enzyme. The enzyme will kind of go around the substrate. It'll bind, right? It goes around it because it's called, we call it induced fit between the molecules to make it fit better, so the chemical reaction can occur better, and then the chemical reaction will happen. All right. Here's another example of it. So you have substrate, right? These go into the enzyme. Enzyme kind of goes around it. Chemical reaction occurs. Products are released. Enzyme is now available again for another reaction to occur. Okay. So that is kind of how that works out. Here's just another couple pictures, right? Catabolism, right? Catabolic reaction where we are breaking down. So enzyme, right, is gray. Green is the substrate, goes in, binds, chemical reaction occurs. And here's an anabolic reaction, which would be the opposite, creating a larger molecule. Just again, another picture, a lot of good pictures here. Substrate goes in, enzyme goes around it, kind of create that induced fit, right? And reaction will occur. So let's talk about one thing that's really cool, right? Enzymes are specific. And what do I mean by that? It's like a lock and a key. You can't use your car key to unlock your house. Only one key will start your car and only one key will open your house, right? It's called the lock and key model or the induced fit theory model, where only one substrate type will fit into one enzyme. Okay, I talked about lactose and lactase. Lactose will only be broken down by lactase. Nothing else will break it down. Okay, so if the shape doesn't fit, it doesn't work. Here's just kind of showing that, right? Like you can see that shape, that round and that triangle has to fit exactly for the chemical reaction to occur. Same as below, right? Oops. So kind of cool, right? That it does have to fit, and that's just kind of how that works. One other thing I want to talk about, because you see this a couple times, the enzyme substrate complex. Okay, what does that mean? It just means the enzyme and the substrate are bound together, and that's when the chemical reaction is occurring. There are things that can affect enzymes. Right? Enzymes don't always work perfectly. There's a lot of things that can affect this. We're going to do a little lab. I'll probably show you a lab on this. But you can kind of see here in these graphs that enzymes work at very specific temperatures or pHs or whatever it might be. Okay, and so we can kind of see here, right? This is a typical human enzyme, right? That works at about 37 degrees, which is your body temperature. That's its optimal zone, right? So you can kind of see, right, optimal zone where it's going to work. If it gets too hot, it doesn't work. Rate of reaction goes down. If it gets too cold, it doesn't work, right? Here's one for pH, right? Pepsin enzyme stomach enzyme, right? Works at about 2, which is good because that's the pH of your stomach. If you get out of that pH, the enzyme doesn't work. Okay, it doesn't change into trypsin over here, right? So because trypsin works at a different one, right? 8 and 2, right? For these different things, and so enzymes, we have many different enzymes to perform many different tasks. Kind of with that, we have many different enzymes because there's also different locations in which chemical reactions occur. Okay, so a lot of different things will affect enzymes, right? pH and temperature and things like that. Different chemicals in your body can affect it too. Some chemicals will actually cause enzymes to not work properly. I'll talk about that here in a second. Okay. And then even like substrate or enzyme concentration. If I have 5 molecules that I need to get reacted on, and I only have 2 enzymes, well, the rate of reaction is going to be slow, right? If I have 5 enzymes, right, it's going to go faster. If I have 10 enzymes, well, it doesn't even go faster than 5 because I just don't have enough to break down, and kind of vice versa as that works out. Okay. So what is actually happening, right? So if you heat up an enzyme or you put in the wrong pH or whatever, you can have what we call denature or denaturation. Okay. Basically, what's happening is enzymes they talk about have a 3D shape. They're folded, right? There's chemical bonds. So all these have like chemical bonds between them, right? And so that kind of holds the enzyme together, right? That gives it its shape, right? Well, if you heat it up, what happens? These bonds are weak, so they break. When they break, the enzyme unfolds. So now you can kind of look, see picture 1, right, is normal. Picture 2 is it unfolded, and now it doesn't work. Okay, that's called denaturing. Right? That's bad news. Why is that bad? Because now that enzyme can't do its job. It can't do what it needs to do. Right? And so we look at that and say, okay, we don't want our enzymes to denature, right? We want them to stay normal. Right? So how do we like keep these optimal temperatures in our body? Right? How do you not overheat? Right? We keep homeostasis. Right? We sweat so our body doesn't overheat, or we shiver to not be cold. So we don't denature. Right? Now the cool thing is this can also go in reverse, and we can get that back, right? Once your body kind of like settles back down. But you could also permanently denature enzymes, which would not be good, right? Because then your chemical reactions would not occur. Just another graph, talk about optimal temperature for different things, right? Don't need that. But here's one that's kind of cool is regulation of enzyme activity, right? So enzymes can be turned on and off, right? Some enzymes, they're not just always working, right? Some actually need to be turned on and off. And so we have what we call allosteric enzymes. Allo, like other site, right? And so we have what we call these allosteric sites on enzymes where molecules can bind to the enzyme to either activate it or inactivate it. Okay, so an allosteric inhibitor will actually inactivate an enzyme. Allosteric activator will activate the enzyme. Kind of makes sense. Okay, which is kind of cool because an inhibitor might be some type of chemical outside your body. Right? If you need, if you like, for example, drink milk, lactose will actually bind to the enzyme and it'll activate it, and you'll start digesting the lactose in your body. Right, well, let's say you are smoking or vaping or something like that, and some enzyme, right, is going to work, right? But all of a sudden, this molecule you inhaled binds to the enzyme and it inactivates it. Well, now your enzymes don't work. Okay, so chemicals like that can actually inactivate enzymes as well, or they could activate it in a wrong way, right? You might be like, well, we don't need to activate this enzyme yet. You ingest something that's not good nor normal. Now you can actually like inactivate or activate your enzymes in the wrong kind of function and use of that. Okay, but also it's also positive, right? Like if you don't just going to create enzymes if you don't need them, your body don't do that. So you'll activate it once you need it as well. Okay, there are other things, cofactors and coenzymes. So like talking about like you need vitamins and these minerals and stuff like that. And that's partly because your enzymes need those to work as well. Some of those are cofactors, right? That'll activate enzymes. And they talk about inhibitors a little bit, molecules that can bind an enzyme to decrease the activity, right? And so we have competitive inhibitors, which will try to bind to the active site. Okay, so again, if you are taking in like food you shouldn't be, or drugs or something like that. These actually might not just be allosteric inhibitors or activators, they could also be competitive inhibitors, okay, where they're actually binding to the enzyme substrate and the chemical reaction can occur, okay? There could be non-competitive inhibitors where something binds to your enzyme, totally changes the shape of the enzyme, and now makes it non-functional. So again, kind of keeping homeostasis, making sure that you aren't ingesting things you shouldn't be is pretty critical when you start talking about enzymes, and then being kind of sensitive to that. Okay, here's just some pictures of it. You can kind of see this is a competitive inhibitor, binds to the active site, so this other thing that's supposed to go in there can't. Here's a competitive inhibition again, so just kind of looking at how different things are affecting enzymes. Okay. Here's another one where you have an allosteric site and it's inhibiting it, so now that enzyme substrate can't work properly. Okay.